Critically shortened telomeres inhibit stem cell proliferation to promote human disease by Dr. Sandy Chang. Dr. Chang is currently professor at Yale Medical School and associate director of molecular diagnostics in the Department of Laboratory Medicine. Dr. Chang's research interests focus on generating novel mouse models to understand how telomere binding proteins protect chromosomal ends from engaging in DNA damage response. Dr. Chang is recipient of numerous awards, including those from the Howard Hughes Medical Institute, the Ellison Medical Foundation, the Sidney Kimmel Foundation for Cancer Research, and the Ellis Benson Award from the Academy of Clinical Laboratory Physicians and Scientists. He was elected into the American Society of Clinical Investigation in 2009. It's my pleasure to introduce Dr. Sandy Chang.
Thank you. Good morning. You guys must have robust telomeres to be up this early to listen to my talk. So my laboratory works on telomere biology, and I want to show you some of our recent data demonstrating that telomeres are actually critically important in preventing the onset of premature aging syndromes and cancer. So if I could get this to work. Okay, so I think I don't have to tell this audience what aging is. You can tell from this picture that the phenotypic consequences of aging is readily apparent to anyone here. But for the purpose of my talk, I just want to define 3 points that leads to the cellular mechanism of aging. So I think we can all agree that aging is associated with the integral and gradual decline of the performance of, for example, organ systems in our body. And aging organ systems are often associated with a decrease in the number of functional cells. And old cells do not maintain and repair tissues as well as younger cells, leading to decreased renewal capacity. For example, stem cells in older individuals have less peripheral capacity than those from younger individuals. Now, the cellular mechanism of aging arose about 60 years ago from classic experiments from Leonard Hayflick. What Dr. Hayflick has shown is that if he takes normal diploid human fibroblasts and just passes them in vitro under controlled conditions, these cells do not have infinite lifespan. In fact, they stop dividing in about 60 to 70 passages, invariably. And this is an intrinsic mechanism, and he was very curious as to why this occurs. You can see from this picture that early passage cells look more ordered. The nuclei are more similar in shape and size. And late passage cells, on the other hand, adopt a very disorganized morphology. The cellular nuclei are disorganized, sometimes they're bilobed, and they adopt a very flat, phenotypically abnormal feature. And this is a hallmark of what we call cellular senescence. Cells actually stop dividing. They're not dead. They still take in nutrients, but they can no longer divide. And we now know that this barrier for cellular division, it actually occurs because we have markers, sequences at the ends of our chromosomes called telomeres, which is the subject of my talk, and they shorten invariably with each round of cell division, and critically shortened telomeres trigger the onset of cellular senescence. And this is the barrier of cell division that Dr. Hayflick have discovered. So what are telomeres? These are structures that caps the ends of almost all eukaryotic chromosomes. In fact, all mammalian chromosomes possess telomere sequences at the ends of the chromosomes. Now, linear chromosomes in all organisms have evolved a method to protect its ends. And in mammals, this is telomeres. These are shown in this green schematic here. And telomeres are TTA-GGG repetitive sequences that terminate in a single-stranded portion that we call single-stranded G overhang. And the reason why this is important is because the enzyme that maintains telomeres is an enzyme called telomerase, which I'm sure most of you have heard about. And this enzyme is a very unique enzyme in that it contains both protein and RNA components. The protein component is called TERT, and the RNA component is called TERK. And the RNA component actually has a sequence that completely is complementary to this 3' single-stranded end of telomeric DNA. So telomerase uniquely is able to de novo generate repeats to telomeric ends in a manner that allows it to progress and lengthen very shortened telomeres. So why do we need telomerase to do this? Now, why did evolution impart this function to an enzyme to maintain telomeric ends. And this is because our DNA polymerase cannot fully replicate lagging strand telomeric DNA. And this is what we call the end replication problem. So during cell division, you get a little gap in that DNA polymerase cannot fill this gap. So in the absence of telomerase, you get whittling of your telomeres with each round of cell division. On the other hand, cells that have telomerase, for example, your stem cells, solve the end replication problem by adding telomeric sequences to chromosome ends. And there, in stem cells, for example, telomeres do not shorten appreciably, and that's why your reserve of stem cells can progress and divide on an almost infinite level because it has the enzyme telomerase. Now, the structure and function of telomerase was recently awarded the Nobel Prize to 3 individuals, Liz Blackburn, Jack Shostak, and Carol Greider in 2009, because these 3 individuals were actually the ones who discovered the enzyme and also its function in normal cellular biology. So this diagram illustrates what happens to your somatic cells without telomerase and germ cells with telomerase. So this diagram plots telomere length on the x-axis and functions of cells in the the telomere length on the y-axis and functions of cells in the x-axis. So as I mentioned, human somatic cells like your skin cells do not have telomerase. This is Dr. Hayflick's fibroblasts. In the absence of telomerase, your telomeres shorten. And when telomeres get to be about 6 or 7 kilobases in length, they're actually recognized as damaged DNA. This is a very important concept. Short telomeres are intrinsically damaged DNA. So this damaged DNA actually triggers a damaged DNA response modulated by a protein called P53, a very important protein that's a guardian of the genome. In the setting of P53 actually recognizes damaged telomeres and tells the cell to stop dividing. So this is actually a very powerful anti-cancer mechanism that humans have evolved. So cellular senescence stops cells with bad telomeres from dividing further. Now, if you're unfortunate to have cells that lose P53, and most of our cancer cells, this is what happens. The cells with short telomeres can continue to divide because this barrier is broached. And short telomeres is actually a very bad and recombogenic. They're very sticky ends. So the chromosomes start sticking to one another. I'll show you some pictures later. It's called chromosome fusions. And the cells enter what we call a period of crisis. Now, this period of crisis is actually another tumor-suppressing barrier. Cells that have fused chromosomes cannot divide very well, and they also die. But cells can actually bypass this crisis by reactivating the enzyme telomerase. And that's what happens in about 90% of all of our human cancers. Telomerase is actually reactivated at this phase, and it selects for cells that can turn on telomerase and have a very unstable bad genome, and this is how we get cancer. Now, 10% of human cells do not reactivate telomerase. They don't use a telomerase base mechanism to maintain telomere length. Rather, they use a recombination-based mechanism called alternative lengthening of telomeres, or the ALT mechanism. Now, these tumors are actually less aggressive than tumors that turn on telomerase. So giving oncologists maybe a means to combat tumors that do not maintain telomeres by activating telomerase. And as I mentioned, germ cells, on the other hand, do have telomerase, and they're basically immortal, although recent evidence have suggested that even in germ cells and our stem cells, telomerase activity is not completely there to prevent critical telomere shortening. So telomeres do shorten over time in humans, even in stem cell compartments. So if you measure telomere length in, for example, fetal liver or fetal brain, their telomeres are pretty long. As I mentioned, you're endowed with long telomeres when you're born. Telomeres shorten with progressive increase in age. You can look at colonic mucosa telomeres. They shorten with increasing age. And in peripheral blood, which is a very good marker to look at telomere lengthening, for example, your human patient population, telomeres do shorten with age. This is shown here in this particular study. Looking at peripheral white blood cells from both males and females, you can see that telomeres shorten very well with age. Studies were conducted with individuals from 60 to about 100 years of age, and the average telomere shortened about, for example, 70 or so base pairs per year. this is what happens to your chromosomes when you have extremely short telomeres. This is a mouse model that I generated. And normally, if you look at a chromosome, which is like, for example, shown here, the chromosome is stained blue, and telomeres are these red dots at the end of the chromosome. So you have 4 dots, at 2 at each end, to protect your chromosomes. So normally, telomeres are very robust. They're very long sequences. They protect at the ends of every chromosome. In this particular mouse model in which telomerase has been deleted, the telomeres shorten. You can see some chromosomes without any telomeres at the ends. And in fact, many chromosomes have short telomeres that are completely missing, and they become very sticky, and they generate the chromosome fusions that I talked about, these end-to-end chromosome fusions illustrated by the yellow arrows. Now, this is obviously a very unstable genome, and this is the type of genome that gives rise to cancer. So I've told you about the role of telomerase at telomere ends. Telomerase is obviously very important in maintaining telomere length, but telomerase is not the whole story. There's a set of 6 proteins that also binds to the ends of telomeric sequences, and they're equally important as telomerase in protecting telomere ends. These proteins are called sheltering because they shield telomeres from the DNA damage response that dysfunctional telomeres invariably generate. And we now know that mutations in these proteins can also generate human diseases that I'm going to talk to you about. So if you have mutations in shelterin, or you disrupt shelterin complex, or if you don't have sufficient telomerase in your cells, the telomeres shorten and become dysfunctional. This is a phrase that I want to impart on you, that we call telomeres that are no longer functional, dysfunctional telomeres. Now, dysfunctional telomeres, as I said, are recognized as damaged DNA. They're recognized just like, for example, if you irradiate cells with IR, gamma radiation, the same type of response are triggered when cells have dysfunctional telomeres. And that is, in the presence of P53, these cells bearing dysfunctional telomeres are actually gotten rid of, either through cellular senescence, they stop dividing, or through active killing of the cells through a mechanism called apoptosis. So as I said, cells bearing bad telomeres are actually fundamentally the human body wants to get rid of, because in the absence of P53, they're actually cancer-promoting mechanisms. So I'm going to talk to you today both about the role of short telomeres in cancer initiation and also in the generation of premature aging phenotypes. So if you look at human pre-neoplastic lesions, like human precursor cancer lesions, these lesions invariably associated with very short telomeres. And this is actually a very interesting and fundamental finding that was recently emerged in the past few years. So for example, if you look at precursor lesions of breast cancer, which is the ductal carcinoma in situ, all these precursor cells have very short telomeres. Same thing with prostatic intraepithelial neoplasia, the precursor to prostate cancer. We could go down the list you can see here. So almost all human carcinomas are associated with short telomeres. And this is also graphically illustrated in this slide here. So this slide shows what happens in the breast ductal carcinoma in situ. This is, again, the early precursor lesion to breast cancer. Now we know that the breast cancer initiating cells arise from the luminal epithelium. And you can appreciate that the luminal epithelium cells contain very, very short telomeres. And telomeres are marked here with a telomapping technique that shows telomeres as these nice red dots. On the other hand, control cells, which is the basal epithelial cells, now these cells do not give rise to breast cancer. They have robust telomeres, as you can see here. So this illustrates very clearly that cancer-initiating cells, such as the luminal epithelial cells, undergo a period of critical telomere shortening before they progress to full frank malignancy. This is another very interesting study looking at patients with inherited cancer mutations in genes called BRCA1 and BRCA2. Now we know that people with BRCA1 and BRCA2, they invariably get breast cancer. This is a very predisposed mutation, a very powerful mutation that gives rise to breast cancer in women at a fairly young age. What was recently appreciated was that these women with BRCA1 or BRCA2 mutations also contain very short telomeres. Now, this is perhaps a correlative finding. Short telomeres correlate with cancer onset. But what was very interesting was that the study also showed that the daughters of women with breast cancer with this BRCA mutation also develop breast cancer, but they develop breast cancer earlier. In fact, patients with daughters develop breast cancers some 5 to 10 years earlier than their mom. And this is what we call genetic anticipation, that daughters of parents with cancer develop cancer earlier. And this earlier onset of cancer is due to the fact that the daughters have inherited even shorter telomeres from their mom. So if you look at this diagram, here's a mom with BRCA1 mutation, and here's her daughter with the same mutation with breast cancer. She has appreciably shorter telomeres and a much earlier cancer onset, typically 5 to 10 years earlier. So short telomeres not only predispose individuals with inherited cancer syndromes to elevated cancer risk, but also the risk occurs earlier in subsequent generation. Now, this finding we have replicated many times even before this study came out. 10 years ago, we've shown this in mouse models of telomere dysfunction, in that when we shorten telomeres throughout the generation, mice with shorter telomeres at later generation develop cancer much earlier than mice with longer telomeres at earlier generations. So these are other studies that correlate telomere length with increasing cancer incidence and cancer mortality. For example, patients with very short telomeres, again, have increased cancer incidence, and also they die earlier from cancer. So the cancers are more aggressive if you have shorter telomeres. This is another set of tumors from a mouse model that I generated several years ago. This is a breast cancer from a mouse model with very short telomeres and no p53. So this model really cemented the fact that short telomeres in the absence of p53 is really a driver for cancer. And you can see that the tumors are very genomically unstable. These are metaphase chromosome spreads showing many genomic instability phenotype. For example, chromosome breaks, fragments illustrated here. This is a chromosome break. And if you do a FISH experiment, fluorescent in situ hybridization, to look at every chromosome in the metaphase spread, you can see many of these chromosomes have generated chromosome fusions. For example, here's a chromosome composed of 3 pieces of other chromosomes, 18, 6, and 5, all stuck to one another. This is a hallmark of human cancers also. Human carcinomas show this type of genome instability, again, due to dysfunctional telomeres. This is again, tumors from the actual mouse model. Breast, skin, and colon cancer are prominent in these mice, and they also show a highly invasive phenotype, for example, dissection into the muscle. Again, this mouse phenotype, the cancer progression and initiation is associated with short telomeres. So this is the mechanism that we think is happening. I think it unifies many of the observations from human models as well as our mouse models of telomere dysfunction, in that if you have epithelium that undergoes continuous renewal, for example, our skin, our GI tract, we renew our GI epithelium every 7 days. And with advancing age, in the absence of telomerase, of course, telomeres shorten down and whittle down due to the end replication problem. This generates chromosome ends that are extremely recombinogenic. They're sticky. They form fusions. These fusions are called dicentric chromosomes. And during a cycle called the bridge fusion breakage cycle, they get pulled to opposite poles of the spindle apparatus during cell division. And these chromosomes can break, and the breakage is random. So they generate double-strand breaks, random breaks, that if they break and destroy, for example, a gene involved in tumor suppression, you can imagine that cells inheriting that mutation will have an increased propensity to develop cancer. And also the breaks can also generate duplications of certain genomic loci, for example, duplications of oncogenes, cancer-causing genes. And that cell and also will have a predisposition to increase cancer. So this telomere dysfunction actually generated like a soup of genome instability. And the soup of genome instability, most of the cells would die. 99.9999% of the cells would die because the unstable genome is actually bad for any cell. But cancer is an insidious disease, and the selection pressure is so great that it will select for the cells that could live with this type of genomic pressure. And in fact, the cells that are selected invariably activate cancer-causing genes, delete tumor suppressors, generating this unstable genome that leads to the progression to carcinoma in situ stage. And again, this is a stage when you could diagnose with very short telomeres. To progress from the carcinoma in situ stage to full frank malignancy requires, again, in 90% of human carcinomas, reactivation of telomerase. So this is actually a critical step, and it's actually a hard step for cancers to do. In fact, many of our cells, carcinoma in situ, do not progress. We don't get cancers all the time. We get cancer with increased age most of the time. And carcinoma in situ is actually, again, another barrier. To reactivate telomerase is not an easy mechanism. But if you can reactivate telomerase, the reactivation of telomerase actually promotes tumor progression. In recent studies around DePenno's lab using mouse models, he's actually generated this entire pathway. He made a mouse with very short telomeres, show that it progresses to the carcinoma in situ stage. And then when he turned telomerase back on in these tumors, boom, they became these huge frank malignant tumors that are extremely invasive with genomic changes that I outline here. So the telomere theory of tumorigenesis is pretty well laid out mechanistically in both mouse and human models. So what about premature aging? I've told you that telomere dysfunction is a DNA damage response that in the setting of a competent p53 pathway generates apoptosis or the cellular senescence mechanism that can lead to defects in highly productive cells like your stem cells, leading to the onset of premature aging phenotypes. And I'm going to illustrate some of this model in the next part of my talk. Again, using mouse models of telomere dysfunction, I've generated mice that actually have stunted growth. These are mice with critically shortened telomeres. You can see that they're smaller than littermate controls with robust telomeres. These are wild-type animals. So this type of experiment obviously is very rigorous because you can only conduct this type of experiment in mouse models in which we could actually delete components of the telomere maintenance pathway or delete telomerase and see what happens to the mouse. So these mice with telomere dysfunction also develop cataracts, they develop hair graying and alopecia, and also skeletal defects reminiscent of kyphosis. And these features are all components of what we think of when we think about human premature aging. So again, a link between telomere dysfunction and the onset of premature aging phenotype, at least in mouse models, has been established. Recently, we looked at what happens in hematopoietic stem cells when you have very short telomeres. And this is the set of experiments outlined here. So if you look at mice that are 2, 4, or 6 months of age, so mice typically live for about 2 years of age. So this represents maybe a quarter of their lifespan, which is relatively early. So what we did was we compared wild-type littermate mice with functional telomeres with mice possessing dysfunctional telomeres. Their sheltering component, a protein called protection of telomere 1, or POT1, has been disrupted, along with telomerase. There's only 50% telomerase in these mice. So these mice have 50% telomerase, which normally they're fine. But coupled with POT1 deficiency, now these mice have very dysfunctional telomeres. And we looked at their hematopoietic stem cells using markers called SCA1 and KIT1 to look at progenitor cells. And these are illustrated by these cells in these 2 boxes. So if you just look at the boxes here. So if you look at wild-type mice that are 2, 4, or 6 months of age, their stem cells are fine. Their stem cells don't change with time. On the other hand, mice with telomere dysfunction immediately at 2 months of age have compromised stem cell function. And the numbers are reduced by 50% compared to wild-type animals. By 4 months of age, there's a 10-fold reduction in the number of stem cells that are present. And by 6 months of age, you can't find any stem cells. These mice have extremely short telomeres. The stem cells are dysfunctional. You can see bone marrow biopsies reveal that they have complete bone marrow failure. So these mice have not only do they have stem cell difficulties, but they are clinically deficient in the generation of bone marrow precursors, resulting in bone marrow failure. If we do telomere fish analysis to look at telomere length, we can actually discover that mice with dysfunctional telomeres and their bone marrow have very short telomeres, and this is marked by this arrow. Telomeres are measured as telomere fluorescent units, and these mice have very short telomeres in that they don't have much telomere fluorescent units as illustrated here, and they have chromosome fusions. Fusion sites have no telomeres, again, all hallmarks of telomere dysfunction. Okay, so I've told you that disease risk in humans actually increases with increasing age. And we all know this. Cancer incidence, the increase in degenerative, for example, diseases, all increases as we age. And we think that in part this is because telomeres are whittled down as we age, And for example, in normal humans, telomeres are so short that when you're 70 or so years of age, that could trigger cellular mechanisms to result in premature aging phenotypes or aging phenotypes. But there's also exists a group of individuals that have short telomeres from the get-go. They've inherited mutations in telomere length maintenance pathways. Unfortunately for them, they have short telomeres as soon as they're born. So the analysis of these patients actually are very informative to inform us about premature aging syndromes due to telomere dysfunction. So I'll tell you about these individuals right now. So if you take the general population of humans and just take their white blood cells and measure telomere length, and we could do this robustly using technologies now. For example, you can see that these are 1, 2, 3, 4 individuals with their telomere length measured. These 3 individuals have telomere lengths that are within the normal standard of deviation, about 10 or so kilobases in length. That's the normal length of our human telomeres. Unfortunately, this individual here, you can see very readily, has very short telomeres. The average telomere length is short, and in fact, the number of very critically short telomeres shorter than 8 kilobase pairs in length, encompass about 80 to 90% of this individual's cells. So this individual not only has short average telomere length, but critically short telomere length in the individual chromosome ends. And if you look at the clinical history of this person actually have bone marrow failure symptoms, very similar to the mouse model that I just showed you. And they also have a mutation in the telomerase gene TERK, which encodes the RNA component of telomerase. So this person actually has a human telomere disease called dyskeratosis congenita. This is a fascinating disease because it's really a first disease of telomere metabolism defect. And these patients are characterized by skin abnormalities, for example, nail dystrophy. They have very abnormally shaped nails because nails, again, are a fast dividing tissue compartment. So if you don't have enough telomeres, you can imagine that this compartment cannot develop or grow very well. They also have leukoplakia, these white plaques in their oral cavity, but they're characterized by early onset of bone marrow failure. In fact, some of these individuals have bone marrow failures as early as 9 years of age, which is pretty sad, actually. These people with dyskeratosis congenita have a 1,000-fold increase in cancer incidence. Again, linking short telomeres with the onset of cancer in humans. They get AML and CLL, and they also have pulmonary fibrosis, which is fibrotic linings in the lung, and which is actually a very lethal phenotype. People with pulmonary fibrosis typically don't live over a year of age. If you measure the telomere length, all the patients with dyskeratosis congenita have critically shortened telomeres. This is a profile of telomere length given in percentiles, and the patients with the disease are illustrated in these red dots. You can see that all of them fall below 1 percentile for telomere length. In fact, most of them fall way below 1 percentile. What's really interesting is that some of these patients have relatives, for example, mothers and fathers, that also have short telomeres, that are illustrated here, but they do not develop the disease, at least not yet. So these are the people that we would like to monitor with telomere testing, because eventually, as their telomere shorten, we think that they will also develop phenotypes of dyskeratosis congenita. Okay, so if you look at telomere length pathways, I've already told you that telomerase is extremely important in maintaining telomere length because it actually generates telomere repeats. The sheltering component is also important. So it's not surprising that mutations in sheltering complex as well as components of the telomerase complex all contribute to the onset of this dyskeratosis congenita disease that I just outlined. For example, dominant mutations in a protein called TIN2, which is a critical component of the sheltering, give rise to a very severe form of disease. And these are the patients that have bone marrow failure as early as, for example, 10 years or so of age. And you can see that many of these enzymes that contribute to the biosynthesis of the telomerase protein can also harbor mutations giving rise to DC. And we recently discovered a set of mutations in a gene called CTC1, which is critically important to involved in telomere replication. So the cells have evolved another set of proteins to replicate telomeres. And you can imagine that if you have aberrant telomere replication, you can also get telomere shortening and disease progression. And we've also found that deletions of CTC1 give rise to a very severe bone marrow failure syndrome. So I think DC patients are very informative in illuminating what can go wrong if you have short telomeres. They just get short telomeres earlier than we do. We invariably will get short telomeres. We will as we age, telomere shortening is an inescapable fact of our life. So what happens to normal humans with short telomeres? What happens is that if you have short telomeres, you are predisposed to having disease risk that are worse than those same age individuals with longer telomeres. This is actually a very famous study that was published in Lancet several years ago, in which you stratified older individuals in terms of telomere length, both in terms of sex and in terms of disease incidence. So you can see that irrespective of sex, individuals with shorter telomeres invariably suffered more disease onset and more fatal disease onset than individuals with longer telomeres. And this correlates, short telomeres also correlates with a 3-fold increase in heart disease and a significant 8-fold increase in infectious disease. So if you have, unfortunately, inherited short telomeres, your disease progression is not going to be great as compared to, for example, a neighbor with longer telomeres with the same biological age. So we now know, and there's a growing literature, that environmental factors can also impact upon telomere length. And I should mention that most of this studies is done in cell cultures, not in humans or in mouse models. And I think those studies are forthcoming. For example, stress, diet, and increased inflammation could result in increased cell turnover. And increased cellular turnover, as we know, is a mechanism for gradually whittling down your telomeres. So if you have exposure to increased environmental factors that are adversely impact upon telomere length, you could potentially reduce telomere length in critically important cellular compartments, generating premature aging syndromes that could arise even earlier than the predicted biological age. For example, we know that from certain literatures that, for example, the compound zinc can actually augment telomerase activity. You can imagine augmentation of telomerase activity could result in at least not an increased shortening of telomeres, but at least a maintenance of telomeres appreciably. And also agents that reduce oxidative stress. And we know that oxidative stress, at least in cell cultures, is actually detrimental to telomere length. Increased oxidative stress have been known to destroy telomeres. You can imagine that compounds that eliminate oxidative stress can also prevent critically shortened telomeres. I should also mention that there are many claims by many compounds to influence telomere length. Now, we have very good ways to test for telomere length, and as a critical consumer, I would urge you to ask companies that are touting their compounds as being pro-telomere length to show the data. To show us the data that your compound actually augment telomere length or maintain telomerase activity, because these studies can be done readily now. Okay, so we all know about personalized medicine in the era of genomic, the various omics, and that will influence our clinical practice. So at Yale, what we're doing is we're actually using whole genome sequencing technology, using next-gen sequencing platforms to actually interrogate every tumor from patients that comes in. At least that's the goal. So we'll sequence every gene from every tumor from every patient with the hope of using this information for personalized medicine, to identify the exact oncogenes, tumor suppressors that are activated or missing in your tumors, and to tailor potential therapies to block or augment certain molecular pathways that maintain tumors. What I think is that this is actually an extremely expensive proposition, not in terms of the sequencing, because the actual sequencing costs of these tumor genomes have come down remarkably, but it's the data analysis that's going to be very labor intensive. And we don't have a lot of the capacity to do data analysis at this point. What I think is that telomere length determination may even be a better mechanism to look in terms of patient care, because it could potentially identify telomere-related diseases. For example, patients with Dysfunction congenita could really benefit from early diagnosis, because then you could do bone marrow transplantation, and that will obviously drastically change their lifestyle. In fact, we're thinking about analyzing unexplained bone marrow failure patients, and by sequencing all the genes that I've illustrated previously that's involved in telomere length maintenance, and the hope of uncovering that some of these unexplained bone marrow failures will actually be due to genetic mutations in telomere length maintenance pathways. I think telomere length determination can also identify healthy individuals with very short telomeres. And those are the individuals that have telomeres that are hovering on the edge of potentially giving rise to disease. And perhaps lifestyle modifications, reducing environmental stress, stopping cigarette smoking, for example, can result in further augmentation of telomere length, at least not further whittling down. And this is also potentially an inexpensive type of intervention that you could do. So how do you measure telomere length? I've already shown you some of the pictures of what I do in the lab, but there's actually 3 different ways to measure telomere length. One is to take cells as a population, hybridize it to a probe for telomeres, and then using fluorescence activated cell sorting or FACS analysis to actually determine the intensity of the hybridization. So the intensity, increased intensity will correlate with increased telomere length. And this is a study that has been very fruitfully conducted in blood samples. But the problem is that it gives you telomere length on a population of cells. So you don't know where the shortest telomeres are in a given cell. You only know your average telomere length in the population. And this doesn't work well with solid tissues. For example, the DCIS picture that I showed you, you cannot use this technology looking at telomere length in precursor lesions. Another method is to use quantitative PCR. This is actually a robust method to actually use PCR-based technology to look at telomere length. Again, this is done in a population setting. The advantage of both of these techniques is that you can actually go through many samples very quickly, so it's very good for large-scale analysis. Again, you can't tell the shortest telomeres in this method in a given cell. So the technique that I use in the lab it's called quantitative telomere fish, which you actually take telomere fluorescent probes, hybridize it onto your chromosome or to your nuclei, and the hybridization signal directly correlates with telomere length. For example, short telomeres will have hybridized to fewer signals, long telomeres will hybridize to more signals. And the cool thing about this technology is 2 things. One is that you could actually detect cells with critically short telomeres. You actually make metaphase spreads and detect even the chromosomes with the shortest telomeres. So that's actually could be very important. You could do this on tissue samples to look at pathologically relevant specimens as to which cell types have the shortest telomeres. And that, as I showed with the DCI sample, is very important. And now there's actually a technique to process large number of samples. So batching these in large-scale analysis is no longer a problem. In the past, this was a technical limitation to this technology. So this technology, again, has the means to look at these critically short telomeres. And looking at critically short telomeres may be even more important than looking at the average telomere population. So if you look here, this is telomere length on a population-based average telomere length. You can see the mean telomere, as I showed many times, decrease with increasing age. But if you look at these individuals and stratify them according to the shortest telomeres, you can see that there's actually a fairly wide scatter. For example, these individuals that are 70 years of age, they have short telomeres, but they're pretty well clustered in a mean short length. But if you look at these individuals in terms of their shortest telomeres, there's actually quite a bit of a variation. For example, this individual here has 50% of their telomeres that are very short. So we now know from Roger Riddell's recent work that all you need is to inherit is 4 dysfunctional telomeres to cause cellular senescence or apoptosis. So only 4 short telomeres in the whole chromosome. So that's enough to trigger the DNA damage response that elicits, at least in cell cultures, senescent phenotype. So monitoring for short telomeres is actually critically important. And short telomeres, again, stratify individuals better than average telomere length. And again, this shows that if you look at your biological age, average telomere length may give you a biological age, for example, of 30 or so years. But if you look at the percent of critically short telomeres, that could actually stratify you as a biological age of closer to, for example, 60 or so years of age. And this is data from an actual person with a chronological age of 66 years of age. If you measure the percent of very short telomeres in this person, less than 3 kilobase pairs, for example, he only has 20% of very short telomere, which is very good. And the percent of medium telomeres or long telomeres is actually in 80 or so percent. So this data suggests that his biological age is actually younger than his chronological age. His biological age is actually about 57 or so years of age, which is almost 10 years less than his chronological age. So biological age and chronological age can be vastly different depending on their telomere status inherited in the individual. So in telomere biology, of course, the goal is to be able to, for example, have developed compounds or methods to increase your telomere length, perhaps to increase the fitness of your stem cells. And this is something that obviously we don't have readily available right now. Many of you are developing compounds that can potentially do this. And I just want to caution that although increasing telomere length may be beneficial, there can also be some adverse side effects. So the beneficial part of increasing, at least transiently increasing telomere length, for example, by augmenting telomerase, is that obviously it could shorten telomere loss. And I've shown you throughout my talk that telomere loss is intricately linked to early onset of cancer and premature aging phenotypes. So if you could stop or maybe delay telomere loss, maybe that could delay some of these phenotypes that I've just outlined. Maybe augmented telomerase function could delay, for example, immune cellular senescence by improving telomere fitness, and also slow down genome instability by reactivating telomerase. This is something that we've done actually in cells in the lab. If you introduce telomerase to genomically unstable cells, telomerase will cap telomeres, maintaining a more stable genome. And also, increased telomerase have now been used in tissue regenerative technologies. For example, at Yale, scientists are making artificial organs using, for example, artificial lungs using lung fibroblasts. And what they're doing with these lung fibroblasts is actually adding telomerase to those fibroblasts so they could live indefinitely and while they reconstitute the lung organ in vitro. So this is actually a very exciting result. So I think regenerative medicine by making artificial organs could benefit from increased augmentation of telomerase function. But the flip side of the coin is that if you add telomerase, as I mentioned before, telomerase in the setting of a carcinoma in situ stage could actually push the cancer to further allow cancer progression. That would be a bad thing. So you don't want to do that. You don't want to just indiscriminately turn on telomerase everywhere in the cell, because would-be cancer cells would be augmented by the addition of telomerase, perhaps giving rise to frank malignancies. So this also has to be done with caution. And if you increase the doubling time of normal cells, there could be increased chance of adding mutations as well. So what about using a telomere length determination mechanism in your clinical setting? And this is something that I do in my practice, that where at least in the laboratory setting, telomere length determination is actually extremely important. And I've outlined some of these here. And I think I've mentioned this several times. Telomere length determination will be critically important to uncover potential patients with telomeropathies, like dyskeratosis congenital patients with bone marrow failure syndromes. I think an unopened field is the use of telomere length diagnostics to look at healthy individuals. Perhaps these are individuals with accelerated telomere shortening, and then that correlates with accelerated biological aging. Even though they're healthy, their telomeres are not healthy, and they may be predisposed to having disease onsets at an earlier rate. And that may lead to interventions in terms of lifestyles. For example, as I outlined, decrease in oxidative damage, decreased environmental stressors, decreased inflammation systemically may lead to reduced telomere loss, which will be a pro-lifestyle change mechanism. And also, in terms of dyskeratosis congenital patients, early diagnosis of critical telomere shortening is actually a life-changing mechanism, because you can actually intervene in these patients. You can actually do bone marrow transplantation using healthy donor bone marrows and significantly improve their lifespan. And these are some of the things that we're actually doing for dyskeratosis congenital patients, bone marrow transplantation. And again, I also mentioned in the future, perhaps tissue engineering, stem cell engineering using telomerase could potentially augment compromised individuals in this regard. Okay, so I just want to sum up my talk, and I hope I've convinced you that short dysfunctional telomeres actually predisposes to increased cancer incidence and increased onset of premature aging syndromes. And I've illustrated that inherited factors, for example, like mutations in telomere length maintenance genes, certain inherited cancer syndromes like the BRCA1, BRCA2 mutations, and things that I didn't talk about like mutations in DNA damage repair pathways, coupled with environmental factors that we know less about, for example, environmental carcinogens, increased reactive oxygen species, and other stressors, all could lead to accelerated telomere shortening, giving rise to dysfunctional telomeres, which are recognized as double-stranded DNA breaks. And the body has evolved mechanisms to deal with double-stranded DNA breaks of any nature, telomere dysfunction being one of them, in that in the setting of p53 sufficiency, dysfunctional telomeres shepherd the cells into cellular senescence, the cells stop dividing, or they die, undergoing apoptosis. And this could, in stem cell compartments, could lead to stem cell exhaustion and the initiation of premature aging phenotypes. On the other hand, if you are unfortunate to inherit, for example, loss of p53, for example, Leaf-Romani syndromes, or you have stochastic loss of p53, for example, in cancer cells or would-be cancer cells, the increased DNA damage resulting from short telomeres is actually a driver for cancer. This is actually really well elucidated now. So short telomeres in absence of p53 drives tumorigenesis, initiates cancer onset, and the increased cancer incidence that we see in mouse models and also in the human population could be largely due to this, the generation and acquisition of unstable genome due to dysfunctional telomeres. And I've also mentioned that telomere length measurements could be an important predictor for increased cellular risk arising from short telomeres. So I just want to thank members of my laboratory who conducted some of the work here. And these are my funding sources for the studies I've outlined in my talk today. Thank you very much.